After being done with the lipid part of the plasma membrane, let us look at the protein component and its functions. These proteins can have diverse functions, and one of the type is called receptors. The receptors can bind with extracellular ligands and then can carry out different intracellular signal conductions. These receptors are usually present on the cell surface, and there are various of them. Then we have channels. Now channels are responsible for transport of different charged ions which cannot cross the hydrophobic lipid bilayer. Now the channels are responsible for transporting along the concentration gradients, while pumps use energy to transport against the concentration gradient. So there are various channels, and as I already mentioned, they are involved in transport of polar molecule like water and charged ions. So the examples can be potassium, sodium, and calcium. And here we have aquaporin, which is specifically for water transport. Now the potassium channel. This is, as the name suggests, can transport potassium, and as we know, in eukaryotic cell potassium has higher concentration inside the cell as compared to outside. Here is a typical structure of potassium channel, which we can see 4 subunits making a ring-like structure, at the center of which we have potassium ion. Now this structure was taken from protein data bank. So this is structure of a prokaryotic potassium channel. Now eukaryotic potassium channel can be voltage gated or ligand gated, which has 4 subunits, each with 4 to 6 alpha helices which span the whole plasma membrane. Now these are the lateral view of 2 prokaryotic potassium channel, and you can see the difference in the structure when it is bound to potassium and it is unbound. Here we have a structure of K+ channel for the ease of understanding. This is one of the eukaryotic potassium channel which is voltage gated. As I said, the potassium channel has 4 subunits, and in eukaryotes we have 6 alpha helices in each of the subunit. Now in this structure we are going to know about the first part of potassium channel, which we refer as the voltage sensor. Now this senses the change in potential and gives a response. Now from the 6 alpha helices, all of them spanning the whole membrane, the alpha helices with the N terminal we refer as the first one, naming it S1, and the one with C terminal we take as S6. So like that we name all other alpha helices. Now the interesting thing is the domain from S1 to S4 is mentioned as the voltage sensor, because this is the one which can sense the change in voltage. How does this happen? So in the S4 we have positively charged amino acid, and where does this positive charge come from? So as I said, there are positively charged amino acid, namely arginine and lysine. Now they have positive charge on their nitrogen atom of their functional group, which gives the positive charge to the S4. Here we can see lysine has a positive charge, and so does arginine. Now these amino acids are present on the 3rd or the 4th position in the S4 helix. What happens is, when there is a change in the electric field, these moves towards the extracellular membrane, this voltage sensor domain, and this causes a change in the conformation, which causes movement of S5 and S6. Now S5 and S6 are the alpha helices which contribute to the pore when we see in the 3-dimensional structure. Now S5 and S6, as they move, the pore will open. Something similar happens when the pore has to close. Like even when there is a voltage change, the S1 to S4 voltage sensor domain will respond to it, and they will move towards the inner cytoplasmic membrane, and this will create a torque in the linker between S4 and S5. Here as you can see, from the S4 alpha helix to S5 alpha helix, we have a linker region. Now the torque in it causes the S5 and S6 to squeeze in and the pore closes. Let us look at the ion conduction pore, which consists of S5 and S6 alpha helix, and we'll come to know why does this pore let potassium ion, a charged ion, pass through the hydrophobic lipid bilayer, and why does it only let potassium pass. So this part has a conserved amino acid sequence which is given like TBGYG. Now T and Y, which are tryptophan and tyrosine, has OH group in their functional groups. You can refer the amino acid chart for it. Now what happens is this gives a structure in the pore something like this, which comes from the 4 different domains. So each domain will contribute one of the oxygen atom through either the O double bond group or the OH group. Now this can stabilize the charged potassium ion to the electronegativity of the oxygen. And also we have this H5 segment connecting S5 and S6. So the oxygen atoms that I showed previously, those are mentioned as the blue dots here. Then we have the potassium ions in green, which are present inside the cell, and they are inside hydration cells or bound by water molecule from every side. What happens, the water molecule cannot pass through the channel because of the presence of oxygen, which act as a filter. It lets only K plus ion pass. So what happens, the water molecule has to let go of the potassium ion, and the potassium ion is called to release its hydration cell. Once it releases its hydration cell, it can now interact with the oxygen atoms of the channel, and then it can get stabilized. Now once it enters the channel, it will have 8 oxygen atoms to interact from all the dimensions. If we look from the top, this will look something like this, and from the side view you can see the potassium ions. This is a 3-dimensional structure prokaryotic potassium channel. The potassium ion gets equally attracted from each of the angle, as it will be interact with 8 oxygen atoms. Now each potassium ion will move further and further through the channel, and